Okay, guys, one more minute. You can hear me well, everyone, right? And you can see the PowerPoint. Everybody, it's 8 a.m. Okay, everyone. First of all, please see that you were supposed to get a metabolic map for MS Exam 3 for vitamins. And I'm not sure, I'm away here, and why it was missed by IT. But MS Comprehensive Exam, you do have a metabolic map. I made sure and I have informed them. Please do, your class rep is here. Please do inform about this metabolic map not available for vitamins. I wish it was, and everyone could have done much better, right? But let's see what we are doing about it. Right now, please see that your MS Comprehensive Exam, since it's a comprehensive biochemistry and genetics, so let's see how we can refresh it. Let me get my laser pointer if I can. First of all, can you hear me well? And are you seeing my slides? Yes. Just a second, guys. Give me a minute. Okay, I'm not missing anything. Great. So first of all, please see there are objectives given to you. Objectives could be the best way to go with this section, but remember that it's a huge section, so it's also good to do a review. I'm not going to go very fast paced, but yes, this is an hour review. So let's see how we do it. Okay.
Recording in progress.
So know that you have biochemistry and genetics both tested on this section because it's a biochemistry genetic comprehensive exam. Metabolic map should be given to your for this exam, and it is there. Let's see what is for starting from biochem, then I'll move to genetics. First of all, you should know acidosis and alkalosis. So what is acidosis? Low pH or high pH? Let this session be a little more interactive. Low pH. Then you have to rule out, is it low pH is acidosis? Then you have to rule out, is it metabolic or respiratory? For metabolic, you see bicarbonate. For respiratory, you see PCO2. Let's do just one, acidosis. Examples then. So very well, very good. Low pH, acidosis. High pH, alkalosis. Then distinguish, is it metabolic or respiratory? That should be your primary. Compensation you already know. When the cause is metabolic, who compensate? Respiratory. But when respiratory is in problem, who compensates? When the pathology, renal. So that's a basic. But first, diagnosis. pH. Can you look at the pH? Diagnose acidosis, alkalosis. Have you done that? Okay. Low pH, you just said acidosis. Then is it metabolic or renal? Or metabolic or respiratory? For respiratory, you see PCO2. For example, chronic obstructive pulmonary disease. Retention of CO2 will lead to what? What is your diagnosis? Chronic obstructive pulmonary disease, retention of CO2. Absolutely wonderful. Respiratory acidosis. Now, metabolic acidosis. Metabolic acidosis, either gain of acid or loss of base. Gain of acid, one example, metabolic acidosis due to gain of acid, anyone example, lactic acidosis. First should come to your mind, diabetic ketoacidosis, because that's more common. Then, yeah, mud piles, absolutely wonderful, but just one example, diabetic ketoacidosis. Loss of bicarb, can you give me example? Is it diarrhea or vomiting? Metabolic acidosis due to gain of acid, excellent, your diabetic ketoacidosis. But metabolic acidosis due to loss of bicarb, is it vomiting or diarrhea? Vomiting would be a wrong answer. Vomiting means you're taking out acid content. So you will be in metabolic acidosis or alkalosis? Vomiting, you would be in metabolic alkalosis. And I was asking you examples of metabolic acidosis. So please don't flip-flop that. Metabolic acidosis due to gain of acid, diabetic ketoacidosis. Metabolic acidosis due to loss of bicarb, diarrhea, not vomiting. So anybody who's saying vomiting is a cause of metabolic alkalosis. right? See, we talked about chronic obstructive pulmonary disease. We talked about diabetic ketoacidosis and also diarrhea, not vomiting. So did you forget that? Where is vomiting here? Do you see it here? Vomiting is what? Do you see my cursor? Metabolic alkalosis. And someone, when I said metabolic acidosis, answered vomiting. That would be a wrong answer. Metabolic alkalosis is vomiting. Metabolic acidosis is diarrhea. Clear? Compensation, I've already told you. When the problem is metabolic, respiratory will compensate. When the problem is respiratory cannot compensate, renal will compensate. Good to know you this reference point. Anion gap is only used to diagnose different causes or differentiate different causes of what? Anion gap is used to differentiate different causes of what? Which disorder? Anion gap is used to differentiate different metabolic acidosis. Absolutely wonderful. Everything clear so far? Yes. Then there was fibrous and globular protein. Please do them. Fibrous do collagen, globular do hemoglobin. Are you aware of deoxy or TOT? Remember, deoxy, TOT, oxy, relax. Which one is because of the making of salt bridges? Deoxy, TOT or oxy-relax, making of salt bridges. When you make salt bridges, which one you stabilize? Deoxy-TOT or oxy-relax? When you make salt bridges, you stabilize deoxy, also known as TOT, or oxy, known as relaxed. When you make salt bridges, which form you stabilize? Deoxy or TOT form. Why is this form important? Because deoxy and TOT form means shifting of the curve towards the right. Does deoxy and TOT form means decreased affinity of hemoglobin for oxygen? And does deoxy or TOT means favor delivery of oxygen to peripheral tissue? Yes or no? Are you aware with all of that? Then you should know factors that shift the curve towards the right or the left. I will finish the left, which were very few. You can reverse the other, but significant are low temperature. Please note down as I'm reviewing. I'll keep my pace a bit okay, right? And R for the review. So again, we are talking about TOT or deoxy and relaxed and oxy. We are concentrating on TOT and deoxy because TOT and deoxy means shift of the curve towards the right. Then you favor TOT or deoxy form. TOT and deoxy form means decreased affinity of hemoglobin for oxygen. TOT and deoxy form means favor delivery of oxygen to peripheral tissue. Now you should know factors that shift the curve towards the right and stabilize deoxy or TOT form. Factors which shift the curve towards the left favor which form? Factors that shift the curve towards the left favor which form? Factors relaxed or oxy form. Let's quickly do factors which shift the curve towards the left. Carbon monoxide poisoning, HBF, hypothermia. Tell me the factors that shift the curve towards the right now. Keep completing. Decrease pH, increase PCO2, very good. High temperature, decrease PO2 and increase 2,3-BPG. Absolutely wonderful. What are the factors that shift the curve towards the right? Low PO2, high PCO2, low pH, high temperature, high 2,3-BPG. Not DNP, 2,3-BPG. There's no DNP here, please. Low PO2, high PCO2, low pH, high temperature, and high 2,3-BPG. They all shift the curve towards the right. Exercise too. Exercise is also lactic acid, low pH. But I'm generalizing these factors. You can add on example. So are you clear with deoxy, TOT, right shift? Oxy, relax, left shift. Left shift were very few, carbon monoxide, hypothermia, and HBF. Remember? Collagen, there were intracellular and extracellular collagen processing steps. You did scurvy, clinical application, and these two collagenopathies. Type 3 was EDS. Type 1 was osteogenesis imperfecta. Brittle bone diseases, which one? Osteogenesis imperfecta or EDS, osteogenesis imperfecta. Stretchy skin, loose joints, that is EDS. They are very clearly distinguished. Type 3 here affected, type 1 here, but they are collagenopathies. Competitive versus non-competitive inhibition. So I'm just touching the points because they are our objectives. So I'm just trying to bring your attention. I hope this is helping everyone. Everyone, it's clear? Competitive versus non-competitive inhibition. Competitive inhibition, inhibitor binds to the same site as substrate binding site. The site inhibitor and substrate compete for that same site. You can overcome the inhibition by increasing substrate concentration. So tell me, what is the effect on kinetics of the enzyme in the presence of competitive inhibitor? Km, increase? Increase, Vmax remains unchanged. How do you distinguish these two from the graphs, whether it's double reciprocal plot or Lineweaver plot, which is linear? In double reciprocal plot, see what is changed. What is changing here? What is increased here on x-axis? What is increased here on x-axis? What is increasing here on x-axis? Anyone? Km, is this competitive or non-competitive inhibition? Competitive or non-competitive inhibition? Competitive. Here, what is unchanged? What is unchanged here? Vmax or Km? What is unchanged here? Vmax or Km? Look at my cursor. Vmax. Is it competitive or non-competitive inhibition? Is it competitive or non-competitive inhibition? Competitive or competitive? And look at this here. What is changing? So in double reciprocal plot, what is changing? In Lineweaver plot, what is not changing? So what is changing here? What is dropping here? What is dropping here in this graph? What is decreasing in this graph? Vmax. Is it competitive or non-competitive? Is it competitive or non-competitive? Is it competitive or non-competitive? What is unchanged here? Km or Vmax? What is unchanged here? Km or Vmax? Is it competitive or non-competitive? Is it competitive or non-competitive? Km unchanged. Km unchanged. Km unchanged is competitive or non-competitive? Anyone? Anyone wants to answer that? Non-competitive. So clearly, competitive versus non-competitive inhibition. You are clear with that. What did we do for energetics? You know that substrate is oxidized, coenzymes are reduced, they go to the electron transport chain and give you ATP. Were there components of electron transport chain? Yes, 5. 4 part of ETC complex, 5th is known as, complex 5 is known as, complex 5 is known as ATP synthase complex. Then we talked about the inhibitors. Some inhibitors inhibit complex 1, 2, 3, 4. Some inhibitors inhibit complex 5 and some were uncoupler. Where did we do carbon monoxide and cyanide poisoning? Complex 4 inhibitor, we clearly identified them. How they are different? Carbon monoxide reversibly binds to complex 4, cyanide irreversibly binds to complex 4. That's why carbon monoxide can be reversed, cyanide cannot be reversed, right? So we identified carbon monoxide cyanide as inhibitor of complex 4. Signaling, you clearly identified lipophilic messenger using intracellular receptor, hydrophilic messenger using extracellular receptors. Did we talk about insulin? You should very well know the well-fed state. What's the sequence of the well-fed state? Do you go to bake sales? Do you eat carbs? When you collect carb, you digest them, you absorb them, and what is released? What is released when carbs are digested and absorbed from pancreas? What is released first in a well-fed state? Insulin. What does insulin do? What is first and foremost function of insulin? To push glucose from circulation into the tissue and turn on its oxidation. And once you have enough of ATP, insulin and ATP, what does body switch from? Catabolism to? Catabolism to, when you have enough of insulin and ATP, storage. What storage is favored by insulin? Lipogenesis. fatty acid synthesis, TAC synthesis, protein synthesis, glycogen synthesis, they are all favored by insulin. So you should clearly know well-fed state versus low insulin fate. But GLUT4 are the only one which are insulin mediated. GLUT4 and GLUT2, they are very important in a well-fed state. Why? Who will tell me? GLUT4 is important because after insulin is released, where is first and foremost insulin going? GLUT4, skeletal muscle and adipose tissue, inside the tissue for utilization. Why is GLUT2 also so important after meals in a well-fed state? What does GLUT2 do? Again, bake cells, digest sugars, absorb them from lumen to cell to circulation, remember? Pick them from transporters. So where does it go first? GLUT2. Why GLUT2 is so important? Why should glucose go into GLUT2? Because where is GLUT2? Where is GLUT2? Where is GLUT2 transporters on the surface of which tissues? GLUT2. GLUT2. GLUT2, anyone? GLUT2. Anybody? Not brain. Brain is 1 and 3. Not skeletal muscle and adipose. You will tell me all of them. Huh? That's not how you do a test, right? You got to be very specific. GLUT2 is liver and pancreas. Absolutely wonderful. That is why it's first very important after a well-fed state, because when you move sugar from lumen to cell to circulation, and circulation, you pick them up. What will sugar going through GLUT2 do in the pancreas? Release insulin. And then insulin pushes glucose from circulation into the tissue. Which one? Skeletal muscle and adipose tissue. So these two transporters are only functional and very important and functional in a well-fed state. So understand GLUT2 and GLUT4. GLUT2 is not stomach and intestine. GLUT2 is not brain and RBC. GLUT2 is liver and pancreas, and GLUT4 is skeletal muscle and adipose tissue. And the only transporters which are GLUT4 mediated which are insulin mediated is GLUT4. So do you understand the importance of these two, GLUT2 and GLUT4, in a well-fed state? They become very active. Though GLUT2 is not insulin mediated, but GLUT2 is very essential to release insulin from pancreas, right? So GLUT2 and GLUT4, absolutely well-fed state, you should understand. The insulin mediated is GLUT4, right? Once glucose we are all doing well-fed state. When glucose is pushed inside, it's turned on for utilization to give you what? Why do you push glucose in a well-fed state from circulation inside the tissue to give you what? To give you what? What do you want? ATP. Once you have good insulin, good ATP, then you will do storage. Whether it's fatty acid synthesis, whether it's TAC synthesis. That's why carbs are killer. When they want to tell you, please, I want to lose weight, physician will first tell you, cut down rice, cut down carbs, because carbs, when you burn carb, you get good ATP, high insulin and high ATP state, then turns on body from catabolism to anabolism. Which anabolism? Fatty acid synthesis, TAC synthesis. So carbs are killers. You got to understand that concept very well. But well-fed state is a state of pushing glucose from circulation to tissue, increasing its utilization. There are many pathways for ATP, but which one is not for ATP? There are pathways for oxidation of glucose. Tell me two pathways which are not for ATP. Glycolysis is for ATP. TCA cycle is for ATP. But there are two. HMP shunt is not. One more. HMP shunt is not for ATP. Mainly, HMP shunt is not for ATP. What is HMP shunt for? NADPH and pentoses. But it's also an alternate pathway for NADPH and pentoses. So every glucose oxidation pathway is turned on after well-fed state, like glycolysis, PDH. Remember, is this a well-fed state? Is PDH well-fed state? VDH, TCA cycle is turned on, but now you have turned on oxidation pathway. Now you have collected enough of ATP. When you are in high insulin and high ATP state, what accumulates in the cytosol? What accumulates in the cytosol, which is a big activator of, no, what accumulates in the cytosol? See my cursor. Citrate that turns on big time. Which pathway that turns on big time? Which pathway? Which pathway citrate turns on? Fatty acid synthesis and taxose. So are sugars fattening? Absolutely. If you go to bake sale, collect carbs, push the sugars from lumen cell circulation, GLUT2, pancreas releases insulin, GLUT4, insulin mediated, both are very essential, key in well-fed state. But once you have turned on oxidation pathway, glycolysis, TCA, HMP is not for ATP, right? HMP is not for ATP. But glycolysis, TCA, now you are in a high insulin and high ATP state, carbs are fattening. They will turn on, they will make citrate available in the cytosol, fatty acid synthesis, and tax synthesis. Also, one more anabolism happens in a well-fed state is glycogen synthesis. Did we do glycogen storage diseases? The disease with morbidity, that means poor lifestyle, is von Gryx disease. Von Gryx disease is because of glucose 6-phosphatase deficiency. So tell me, what will you see in von Gryx disease? What would be the primary presentation of von Gryx disease? Which is not seen in Pompey's, which is not seen in McArdle's. Von Gryx, severe fasting hypoglycemia, lactate accumulation, hyperuricemia. It's a myriad of symptoms. But first and primary is, yes, enlarged liver because of deposition of what? Why is the liver enlarged? Why there is hepatomegaly? What is not being used? Then glucose 6-phosphate glycogen, absolutely wonderful. So we did glucose 6-phosphatase, von Gryx disease. Pompey's does not have hypoglycemia. It's a lysosomal storage disease. Keep an eye on your lysosomal storage disease. Can you broadly give me which are your lysosomal storage disease? Broad groups, not example. Glycogen storage, Pompey's. What else? Give me your lysosomal storage diseases. Glycogen storage disease, Pompey's, mucopolysaccharidosis. Somebody is saying Hurlers and Hunters. Absolutely wonderful. Mucopolysaccharidosis. What else? Somebody said Tay-Sachs, sphingolipidosis. I'm bringing the broader classification, right? Glycogen storage disease, Pompey's, Hurlers, Hunters, mucopolysaccharidosis, sphingolipidosis, Tay-Sachs, Fabry's. And then we have, Gaucher's too. They're all sphingolipidosis. I'm talking about bigger groups. Glycogen storage disease, Pompey's, mucopolysaccharidosis, Hurlers, Hunters, sphingolipidosis. I will bring all of them. Everybody's giving right example, but go broader up. And the last one, can anyone tell me a last lysosomal storage disease? Anybody? Anybody can tell me a last which we are missing still? Eye cell disease, I like that. Well, hexosaminidases, Tay-Sachs, that is in sphingolipidosis, right? Eye cell disease is a bigger group. So eye cell disease, sphingolipidosis, mucopolysaccharidosis, and glycogen storage disease, Pompey's. Right? Look at them. So we talked about McArdle's. McArdle's does not have hypoglycemia. This is decreased exercise tolerance. So see, is von Gryx different from McArdle's? You can't flip-flop that. Absolutely different, right? Does that make sense? Now, HMP shunt is not for ATP. What is it for? NADPH and pentosis. And what is NADPH for? What does NADPH do? Does it maintain RBC integrity, neutralize hydrogen peroxide in RBC membrane? So when there is a G6PD deficiency, will you see low NADPH? Will you see low NADPH? Glutamine? There's no glutamine. Okay, sorry. When you see G6PD deficiency, low NADPH, what will accumulate in the RBC membrane? Glutathione. Yes, glutathione will not be functional. What will be reactive oxygen CC, hydrogen peroxide, and hemolytic anemia? Here, hemolytic anemia is because of predisposing factor. Yes. We also did nitric oxide. Is it an endothelium derived? No worries. I'm also quick and you're also quick, right? You want to finish calm and just be over it. I can understand that. So when you talk about nitric oxide, it's an endothelium-derived relaxation factor. Remember, I told you to make a list of amino acid and what they form. Arginine forms nitric oxide in your body. And what does tyrosine form? Arginine forms nitric oxide. What does tyrosine form? Tyrosine, melanin, catecholamines. So two things you can learn from here. Nitric oxide is an endothelium-derived relaxation factor and arginine forms them. Here somebody was telling me lysosomal storage disease, hurlers and hunters, which is X-linked. Hurlers or hunters? X-linked. Hurlers or hunters? Hunters. Hunters hunt for an X and they have a good eye, right? Eye cell disease was lysosomal targeting defect. Then we did fatty acid. Why do we do fatty acid in a well-fed state? Because carbs are fattening. First, you go to bake sale, collect carbs from, digest them, absorb them from lumen to cell to circulation. Then they go to the glucose transporter. GLUT2, pancreas release insulin. GLUT4, insulin pushes glucose inside skeletal muscle and adipose tissue. Insulin well-fed state is also a state of glucose oxidation except HMP pathway. But now when you have enough of insulin and ATP, you got to do fat metabolism, and especially which metabolism? When you are in high insulin and high ATP state, which fat metabolism you want to turn on? High insulin and high ATP, tell me. Which fat metabolism pathway you want to turn on? High insulin and high ATP, fatty acid synthesis. But before that, bile is important for absorption of fat. Bile is important for digestion. Bile is important for absorption, micelle formation. And that's why there are two stimulus called cystokinin, CCK, and secretin. Which one helps in stimulating bicarbonate-rich pancreatic fluid? Cholecystokinin or secretin? Bicarbonate-rich pancreatic fluid is secretin. So you can flip-flop their roles. Not cholecystokinin, please. Secretin helps in bicarbonate-rich pancreatic fluid. So understanding their first fatty acid synthesis, you got to see how do you absorb and digest and absorb fat. So bile, role of CCK, secretin, fatty acid synthesis, well-fed state, and this is a great stimulus for fatty acid synthesis. This comes when you are in high insulin and high ATP state. That's why sugars are fattening. What does citrate turns on? Big time fatty acid synthesis. So fatty acid synthesis is the well-fed state pathway. Is fatty acid synthesis a well-fed state pathway? Yes, but when you are in high insulin as well as high, fatty acid synthesis is when you are in high insulin as well as high, what? Citrate plus ATP, right? That's why sugars are fattening. So you got to understand this concept of citrate pushing fatty acid synthesis. That is why carbs are fat. Just as citrate is so essential for fatty acid synthesis, who will tell me what is essential for fatty acid oxidation? Without that, you can't push fatty acid from cytosol into the mitochondria. You cannot. Carnitine and carnitine shuttle. Is carnitine source animal diet, animal food? Is animal food source of carnitine? Is animal food source of carnitine? Right? Yes. So first understand the key players are coming. See, I'm not going into details of every enzyme, right? How I'm talking about broad. When I'm doing fatty acid synthesis, are carbs fattening? Is it in high insulin as well as high ATP state? What is fatty acid oxidation? Key carnitine, animal food. Now this pathway is turned on, HSL. Is turned on big time in diabetic ketoacidosis, especially diabetic ketoacidosis is type 1 or type 2. Diabetic ketoacidosis seen in type 1 or type 1 absolutely turned on this pathway. So naturally tag lipolysis, then ketogenesis and gluconeogenesis, that's not a well-fed state pathway, right? In this case, this is altered metabolism. So this big TAP, HSL, is turned on in high insulin or low insulin? This high HSL and tag lipolysis and then subsequently low insulin state. That's why what's the primary treatment of type 1 diabetes? to shut down everything, to shut down the HSL, to shut down the fatty acid, gluconeogenesis, ketogenesis, insulin. Insulin is a primary treatment of type 1 because of this only, to turn off HSL. So this is also one clinical application of carb disorders. But HSL active should also be related to well-fed or low insulin state. HSL active should be related to well-fed or a low insulin state. Low insulin state, you should be clear by now. You can't flip-flop that. Yes, remember the lysosomal storage disease we talked about, Pompey's, glycogen storage disease. Now this one, this big is sphingolipidosis. Mucopolysaccharidosis, I already did, Hurlers and Hunters. Eye cell disease, I already did, lysosomal targeting defect. But look at this, the disorder and the enzyme. Somebody said, somebody says hexosaminidase A, Tay-Sachs, Fabry's galactosidase, only one X-linked, Gaucher's glucocerebrosidase, Niemann-Pick sphingomyelinase. These were all sphingolipidosis. The only X-linked was which one? Fabry's. So there are specific disorder with specific enzyme. Did you pay attention on that? Right? And understand that intracellular cholesterol pool is very important to maintain LDL on the surface, right? On the surface of the liver. If your intracellular cholesterol pool is too much, obesity, LDL receptor function can be lost on liver surface. Or there could be familial hypercholesterolemia where LDL receptors are defective, and that's not good. That's atherogenic. You are aware of that. Did we do 21 versus 17 alpha hydroxylase deficiency? Are you following my pace? And tell me when it's an hour. I think it's 30 minutes over, right? I'm going at a good pace. I don't want to hold you. I want to tell you the key points. You have time to review them, right? Are you following my pace? Is that helping? Everybody? Great. Now you compare 21 alpha with 17 alpha, clearly distinguishable. You see virilization of female and hypotension. Where? Yes, I know we have time. Virilization of female and hypotension in 21 alpha, whereas reduced sex hormones and hypertension in reduced sex, no, somebody is saying 17, no. Virilization in female and hypotension is 21 alpha, please. Virilization in female or masculinization of female external genitalia and hypotension is 21 alpha, more common. But hypertension and low sex hormone, yes, hypertension I'm saying. Don't say 11 beta. I'm saying hypertension and low sex hormone. I'm clearly distinguishing 21 alpha from 17 alpha. Virilization of female external genitalia and hypotension, 21 alpha. Hypertension and low sex hormones, 17 alpha. Eicosanoids, 17 alpha. Absolutely. They are clearly distinguishing. Eicosanoid has cyclo-lipoxygenase pathway. Aspirin is a cyclo-inhibitor or lipo-inhibitor. Aspirin, indomethacin. What are they? They are cyclo-inhibitors or lipo-inhibitors. Cyclooxygenase inhibitor or lipo-oxygenase inhibitor? Cyclooxygenase inhibitor. Urea cycle in the liver, 2 mitochondrial steps, 3 cytosolic path. Amino disposal is important for amino acid. Now we have come to protein and amino acid. Amino disposal is important because ammonia is dangerous, toxic, diffusible gas. But remaining carbon skeleton is not a waste, right? That's why you have a glucogenic amino acid, alanine, which are ketogenic amino acid. Glucogenic amino acid is alanine, which are ketogenic. Ketogenic are amino acids are ketogenic. Ketogenic amino, leucine and lysine. Ketogenic are leucine and lysine, right? Glucogenic is alanine, but there are some conditionally essential amino acid. In PKU, what becomes essential? Tyrosine. What becomes essential in urea cycle enzyme deficiency disorder? Conditionally essential means they become essential only under certain condition. So in PKU, tyrosine becomes essential. Otherwise, it's not essential. When does arginine become essential? When does arginine become essential? Arginine. Urea cycle defect, growth, reproduction. Yes, urea cycle defect. Arginine becomes essential. Is arginine formed from urea? From urea cycle? Yes. Arginine is used for nitric oxide production. That's the second step. But I'm asking, when does arginine becomes essential? In urea cycle enzyme defect. Yes, because urea cycle produces arginine. So some amino acids are conditionally essential. Why folate is so important for purine ring, pyrimidine, and methionine synthesis? Remember, because folate deficiency, low folate in diet, megaloblastic anemia. I don't know how many times I've said that. I hope you did that well on the test. B12 is folate trap, also megaloblastic anemia. But unfortunately, I don't blame you. The metabolic map should have been provided to you. And as I've told you, that was a requirement. Let's move on. Let's move on to our comp. Do it well. So folate is required for purine pyrimidine synthesis, folic acid deficiency, low folate in diet, megaloblastic anemia. B12 deficiency, folate trap, again megaloblastic anemia. PK classic is enzyme or cofactor defect? Classic is enzyme or cofactor defect? Classic is enzyme or cofactor defect? Classic is enzyme. So what is high in PKU? Classic PKU. What is high and what is low? What is high and what is low in classic PKU? What is high and what is low in classic PKU patients? Anything? Anyone? What could be high? Phenylalanine is high. And what is low? Tyrosine is low. Absolutely wonderful. Absolutely wonderful. In classic PKU, is there anything wrong with cofactor? In classic PKU, is there anything wrong with cofactor? No. So classic versus non-classic. You clearly identify, this is the defect P in PKU. Phenylalanine cannot be converted. This is normal. This is abnormal. This is PKU. Phenylalanine cannot be converted into tyrosine. That is why we have some conditionally essential amino acids. Tyrosine is conditionally essential. PKU patient. Arginine is conditionally essential. Urea cycle enzyme deficiency disorder patients. Right? Did we clearly do purine versus pyrimidine? De novo versus salvage? Catabolism purine or pyrimidine, which is more dangerous? Catabolism purine or pyrimidine, which is more dangerous, like which is more alarming clinically? Catabolism of purine or pyrimidine? Excess catabolism of purine? Purine, because of hyperuricemia and gout. Did we do de novo as well as salvage? Where is PRPP amidotransferase rate limiting enzyme? You should know rate limiting enzyme. Where is PRPP amidotransferase rate limiting enzyme? De novo purine or de novo pyrimidine? De novo purine or de novo pyrimidine? Where is PRPP amidotransferase rate limiting? De novo purine. CPS1 is rate limiting for? CPS1 is rate limiting for? CPS1 is rate limiting for? Anyone? CPS1. CPS1. You're forgetting CPS1. You're forgetting CPS1. CPS1. I'm not saying CPS2. CPS1. Urea cycle. CPS1 is urea cycle. Remember urea cycle, right? You should know rate limiting enzymes. Some of the pathways I have addressed. I'm giving you key points. You know well-fed versus low insulin state. Well-fed state is high insulin. When you're in high insulin and high ATP state, body switches to anabolism. That's why carbs are fattening. What anabolic pathway are switched on when you're in high insulin, high ATP state? Fatty acid synthesis. TAG synthesis, glycogen synthesis, or protein synthesis, right? Are carbs fattening? Absolutely. Is this the sequence? You go to bake sale, you collect carbs, digest them, absorb them, push them from lumen to cell to circulation. Where do they move in blood to? Pancreas. What is released? What is released? Pancreas. Release what? Insulin. Is this all well-fed? Absolutely, it's well-fed. And then what's insulin function? Push glucose from circulation to tissue. Only one insulin-mediated GLUT4. GLUT4 are active after a well-fed state only. And also insulin turns on oxidation, and well-fed state is a state of glucose oxidation. Once you have enough ATP, then you do anabolism. So insulin has a big role to play. GLUT2 has big role to play after well-fed state. because pancreas release insulin. GLUT4 have a big role to play. They are active only in a well-fed state. What is GLUT4 doing? Insulin quickly pushes glucose through which tissue? GLUT4. Insulin quickly pushes not only just skeletal muscle but also adipose tissue. So insulin is a well-fed state hormone. Insulin is a well-fed state hormone, whereas glucagon, epinephrine, and cortisol are low insulin state. You clearly know that, right? So in a well-fed state, you will turn on glucose oxidation. When you have enough insulin and ATP, fatty acid synthesis and TAC synthesis. So are carbs fattening? Absolutely fattening. Carbs are fattening. This is all well-fed. On the left is well-fed. On the right is low insulin. I'm not doing low insulin right now. I'm doing well-fed. What else is well-fed? Protein synthesis is well-fed. You know clearly. Low insulin. Low insulin, please understand, distinguish where glucagon. Tell me. Low insulin can be high glucagon, high epinephrine, high cortisol, right? But now you should know which pathway primary, which hormone. So tell me, glucagon, one primary pathway it will turn on. Glucagon, one primary pathway it will turn on. Give me one. One and the first and the foremost that comes to your mind. Glucagon is low insulin. Not. See, glycogen lysis is a bad answer. Glucagon, gluconeogenesis, absolutely wonderful. Epinephrine, give me one primary pathway that is turned on. Give me one primary pathway. First, list first. Epinephrine, give me one primary. They are all low insulin. Glucagon, gluconeogenesis. Epinephrine, anyone? Big one, epinephrine. Flight, fight, and fright, quick energy. Which pathway? Quick energy. No, gluconeogenesis, wrong answer. Glycogenolysis, glycogen breakdown. Glucagon, gluconeogenesis. There can be a big list. I want first, okay? First pathway first. Absolutely wonderful. Epinephrine, glycogenolysis. Glucagon, gluconeogenesis. There could be many. I'm talking about primary. First, that should come to your mind. Cortisol, gluconeogenesis or glycogen breakdown? Cortisol, gluconeogenesis or glycogen breakdown? Cortisol, gluconeogenesis or glycogen breakdown? Glycogen breakdown, wrong answer. Gluconeogenesis, because cortisol takes a lot of time. It needs an intracellular receptor. Glycogen breakdown is flight, fight, and fright, quick energy. Glycogen breakdown, absolute. No. Who's saying glycogen breakdown? I'm saying, what did I ask you? Cortisol is a no for glycogen breakdown, please. Wrong answer. Cortisol primary, gluconeogenesis. Did anyone answer gluconeogenesis? I think nobody, right? Be mindful. Glucagon, gluconeogenesis. Epinephrine, glycogen breakdown. Cortisol, gluconeogenesis. Cortisol primary, glycogen breakdown, wrong answer, right? Then HSL. Out of all these three, which one you want to put HSL? Give me two. Out of three, two are very important for HSL activation. Two, only two. Glucagon, wrong answer. Glucagon, wrong answer. For HSL, it's epinephrine and cortisol. Did you forget that? Glucagon, you don't talk at two places. Glucagon is not for muscles. Glucagon is not much for adipose tissue. That doesn't mean it's an absolute no. Primary. You should know my primary hormone, my primary pathway. So when I'm saying HSL, is it adipose tissue lipolysis? Is HSL adipose tissue lipolysis? HSL activating adipose tissue lipolysis? Yes or no? Tag lipolysis. HSL, is it tag lipolysis? No glucagon there, please. Epinephrine, cortisol, HSL. Cortisol, gluconeogenesis. Cortisol activating HSL. Epinephrine activating glycogen breakdown. Glucagon activating gluconeogenesis. Some of the key pathway don't flip-flop. That's why see hypoglycemia, what is released? That's why glucagon supports big time gluconeogenesis. When I asked you glucagon, your first answer should be gluconeogenesis. Flight, fight, and fright, epinephrine, glycogen breakdown. Cortisol, gluconeogenesis, right? Type 1 and type 2. So do you understand the primary two? Please don't flip-flop the primary ones. If you have any questions, I'll give you a few minutes later when we finish. Type 1, type 2. Why type 1 is so insulin dependent? Because type 1 has severe insulin deficiency. HSL is turned on big time. And there is too much fatty acid, too much gluconeogenesis, too much ketogenesis. Why do you give insulin as a primary treatment in type 1? To shut off HSL. That's why you give type 1 diabetes. So type 1 are primarily dependent on insulin. You know only endogenous insulin has C-peptide, only endogenous, not exogenous. There are other types of diabetes too. Why MODY, which is diabetes of young, so clinically important? It is related to which transporters, tell me, and which enzyme. That's why I told you in well-fed state, two transporters you need to understand well. Well-fed state, then they are very active and functional. So maturity onset of diabetes is related to which enzyme and which transporters. Which transporter enzyme you need to know to understand maturity onset of diabetes of young, not GLUT4, not GLUT4, GLUT2 and glucokinase. Very good. Somebody said GLUT2 and glucokinase. Glucokinase defect is maturity onset diabetes of young. This is not for you to now go back, do a maturity onset diabetes of young, but look at the transporters that you need to understand with well-fed state, GLUT2, GLUT4, glucokinase. They are all well-fed state. They have to be for them to be active, you got to be in well-fed state. You get my point? For now comes the genetics. You know the central dogma. You know the replication. So leading strand or lagging strand? Where do you see Okazaki fragment? Where do you see Okazaki fragment? Lagging strand. So these are the basics. Understand them. Go back and see them. You did single-stranded and double-stranded break repair. We did transcription. Transcription is forming mRNA, tRNA, and ribosomal RNA, right? Remember, transcription is not just complete by RNA polymerase. There is co- and post-transcriptional processing. Then they are complete. Like transcription, translation, they all have initiation, elongation, termination. But remember, so we did replication. Each strand acts as a template. There's a leading and lagging strand, Okazaki fragment lagging strand. Then we did transcription. Transcription is different from replication. Only one strand acts as a template. You synthesize mRNA, ribosomal RNA, transfer RNA. You need RNA polymerase. Transcription and translation, each one of them have initiation, elongation, termination. Transcription is not complete until you do the co- and post-transcriptional processing. Remember, that is transcription. Yes, I'm just bringing the key points. So read them in the same context. Don't get absorbed in it. Remember, it's biochemistry and genetics, full volume. So I'm just trying to give a bigger picture. So yes, transcription, only one strand acts as a template. Only one strand acts as a template. What do you synthesize? Ribosomal RNA, mRNA, transfer RNA. What enzyme you need? RNA polymerase 1, 2, and 3. Are there cis and trans elements? What trans factors? So trans factors are not part of DNA. But what are the trans factors, do you know, Kelvin? B, D, F, H. What was B? A bridge between F and H. What was D doing? It binds to DNA. What is F doing? It recruits RNA polymerase. What does H do? It activates RNA polymerase. Why do you need RNA polymerase? Because you want to do transcription. Here you will not use DNA polymerase, you will use RNA polymerase. Genetic code, triplet, continuous, degeneracy, and mutation. Mutation are very important for genetic code. So how many mutation we did? Remember, we did point mutation, we did trinucleotide repeats, we did frame shift, we did non-frame shift. Give me one example of point mutation. One example. Point mutation can be silent, missense, and nonsense. Silent, no change in amino acid. Missense, change in amino acid. Nonsense, stop codon. Give me one example of point mutation. One example of point mutation, sickle cell disease, absolutely wonderful, very good example. So you should understand some examples of point mutation. Now give me example of one trinucleotide repeat expansion, just one trinucleotide repeat expansion, just one of them, trinucleotide. So you understand, no, this is point mutation. Is this missense, silent or nonsense? Then you identify. Huntington's disease, trinucleotide. Then there was frame shift and non-frame shift. Where was cystic fibrosis? Frame shift or non-frame shift? Frame shift or non-frame shift? Cystic fibrosis, frame shift or non-frame shift? Non-frame shift, deletion of 3 or multiple of 3 with no change in amino acid at either end, N or C terminal, non-frame shift. So for genetic code comes important is these mutation, which is your point, silent, missense, nonsense. Then your trinucleotide repeat, then your non-frame shift and frame shift. Cystic fibrosis was clearly a non-frame shift mutation, right? Yes, do you remember that? Yes. Translation was initiation, elongation, termination. Initiation is complete when small subunit, large subunit, mRNA, transfer RNA come together. Elongation is elongating the peptide chain. Termination is releasing the peptide chain. Right? We did their basic principles. It also has initiation, elongation, termination. See how I'm going a little broader, right? Not getting into details, dipping and dipping. I'm just warming my feet, not soaking them in. So you do the same. You know what is sense, what is template. mRNA is same as, except T is U. mRNA sequence is same as sense or template. mRNA is sequence is same as sense or template, sense or coding. Then you have codon and anticodon arm, which is required for protein synthesis. Did we talk about cis and trans elements? Cis are part of DNA, trans are not part of DNA. Trans factors are those trans elements which are not part of DNA. In trans factors, you did BDFH. Remember, you did BDFH. Trans factors do bind to DNA, but they are not part of DNA. Part of DNA are known as cis elements. Did we clearly identify that? Right? Inheritance pattern. We did autosomal recessive, X-linked recessive, autosomal dominant, and mitochondrial. Autosomal recessive. Do both parents need to carry the mutant allele? Do both parents need to carry the mutant allele? Do you need 2 mutant allele to express the phenotype? Do you need 2 mutant allele to express the phenotype? Are phenotypically normal means both parents are carrier. The phenotypically normal couple has a autosomal recessive phenotype in a child. Are both parents carrier? So do both parents have to carry the mutant allele? Absolutely in autosomal recessive disease, right? When you go to X-linked recessive, only one parent has to carry the mutant allele. Who's that parent? Mom and who can that be? Who can be a carrier here? Mom and not dad. Not in X-linked recessive. Dad is not a carrier. Mom and daughters can be carriers. Sons are affected. In autosomal dominant, you only need 1 mutant allele to express the phenotype. So do you talk about carriers in autosomal dominant? No. Okay. So autosomal recessive, both parent needs to carry the mutant allele. You need 2 mutant allele to express an autosomal to express the phenotype in an offspring. If both parents are phenotypically normal for autosomal recessive, they are carriers. X-linked recessive, one parent can be carrier, that is mom and daughter. In autosomal dominant, you just need 1 mutant allele. You don't need any care. You don't talk about carriers there. How do you identify them? Is this autosomal dominant or recessive inheritance pattern? So you should also be able to diagnose this. Is it autosomal recessive or dominant? This one. It's dominant. There's no skipping of generation. Male and female equally affected. That is autosomal dominant. Autosomal dominant and recessive, male and female are equally affected, right? Yes, this is absolutely an autosomal dominant inheritance pattern. You need 1 allele, 1 mutant allele to express the phenotype. Is this autosomal recessive now? Sorry, too many windows. Yes, this is autosomal recessive, skipping of generation. Yes. And why they are obligate carriers? Because they have an autosomal recessive phenotype offspring. So they are obligate carrier given by TOR. Is there a 2/3 rule? Yes. 2/3 rule is for autosomal recessive disorder. Both parents have to be carrier and the sibling they are asking should be unaffected. So 2/3 rule will come with which disorders? 2/3 rule should come with which disorder? Autosomal recessive. In autosomal recessive, do both parents need to carry the mutant allele? Absolutely. It's not possible that one parent carries the mutant allele. One parent carrying a mutant allele is possible in X-linked recessive or autosomal dominant. Is this 2/3 rule? 2/3 rule, you should know, autosomal recessive, both parents should be carrier, sibling they are asking is unaffected. Now, what is this disorder? Is this dominant or recessive? Is this dominant or recessive, first of all? Is this dominant or recessive? Dominant recessive. What is it? X-linked recessive or autosomal recessive? X-linked recessive or autosomal recessive? X-linked, because males are affected. Males are more affected. It's an X-linked recessive disorder. Absolutely wonderful. And what is this pattern? This should not be missed. First, always rule out mitochondrial. If mom is affected, all children are affected. If dad is affected, none of the children is affected. What is this disorder? Don't confuse this with X-linked. Mom affected, all children affected. Dad affected, none of the children affected. Then examples, also example. So is PKU mitochondrial? Is PKU mitochondrial? So what is PKU? What is PKU? Inheritance pattern. Some example you should know. PKU. PKU. Inheritance pattern. PKU. PKU. Autosomal recessive, achondroplasia, Marfan syndrome. PKU, autosomal recessive, achondroplasia, Marfan syndrome, autosomal dominant. So some example also, I've given it in the review PowerPoint. Look at this. Can you clearly identify what is this inheritance pattern? Skipping of generation, double line, autosomal recessive. Yes. Is this autosomal recessive inheritance pattern? Yes. Absolutely wonderful. And then please understand the 2/3 rule. You know the 2/3 rule too. Oh, sorry, always unaffected sibling of an affected individual, both the parents need to be carriers, right? What is this disorder? Again, there is skipping of generation. Are male more affected than female? Are male more affected than female? It's an X-linked recessive disorder, right? It's an X-linked recessive disorder. You can clearly identify that. Is this mitochondrial? Is this mitochondrial? Is this mitochondrial? Why not? In mitochondrial, what happens? What happens in mitochondrial? What happens in mitochondrial? If mom is affected, all children are affected. It's not dad, don't give it. If mom is affected, all children. How do you identify in a pedigree, I'm asking? If mom is affected, all children are affected. If dad is affected, none of the children is affected in mitochondrial disorder. Microarray, there is a DNA microarray, there is a cDNA microarray. Simply, what is the color of the control here? What is the color of the control here? What is the color of the control here? Green. And what are they asking? Deleted or amplified? What are they asking? Amplified. What is your answer? What is your answer? First color of the control, amplified. Your answer is? What is the number? Yeah, give me the number. 2 and 4. Absolutely wonderful. Nothing fancy in this. Let's go further. Okay, cDNA. Same thing. Color of the control. Color of the control is what here? What's the color of the control here? What's the color of the control here? Green. And what are they asking? What are they asking? Upregulated. What is your answer? What is your numbers? What are your numbers? Color of the control is green, upregulated. What are your numbers? 2 and 4. Absolutely wonderful. Nothing fancy about it. Karyotype. Karyotype, fish and chromosomal painting. They can identify anything. Aneuploidies, autosomes, sex chromosomes, chromosomal translocation. So tell me chromosomal translocation, fusion gene, gene under strong promoter. Give me fusion gene examples. Fusion gene examples. CML and CML is which chromosome? CML is which chromosome? 922. What is the other example? CML and PML or AMLM3. What is PML, AMLM3? 15, 17. What was gene under strong promoter? Burkitt's and follicular, right? So can you identify by these pattern? Absolutely, why not? By karyotype, fish, chromosomal painting, you can identify them. This is clearly CML. What chromosome do you see in CML? See, is this fish? This one was karyotype and this one is fish. Is this fish? So why this is fusion gene? Because red and green gave you what? Red and green gave you a color yellow. Yeah. So this could be either CML or PML, known as AMLM3, but this is clearly CML. What's the chromosome? Philadelphia chromosome. Population genetics. P squared is autosomal dominant. Q squared is homozygous autosomal recessive. 2PQ is carrier. I told you, how do you go about the question? First, identify the inheritance pattern. Is this autosomal recessive? Is this autosomal recessive? So what's 16%? What's 16% here? What's 16% here? Q squared, P squared. Q squared, P squared, Q squared. And what are they asking you? What are they asking you? What 2PQ? But don't forget times. Don't forget times what? Times. Sorry, I'll correct that. It's 26,000. 6,000, right? I think I made that correction. I don't know. X-linked, an exception. Here, incidence of disease is Q squared or Q. But here it is given albinism's gene. Sorry, this is not X-linked. This is autosomal recessive. This is autosomal recessive. But they have given not incidence of disease, but or frequency of disease, but allele frequency. So here you will say it's Q, but otherwise, autosomal recessive, incidence of disease is what? Incidence of disease is what? Autosomal recessive. Incidence of disease is Q squared. Unless they say gene frequency, then it becomes Q. Otherwise, it will always be Q squared, right? Here, they are not asking carrier. They have given you a pedigree and they're asking what is the probability that the child will be affected, right? Does that make sense? Right? It's different. But autosomal recessive, please always assume that the incidence of the disease is Q squared. Then whatever is given, first inheritance pattern, then is it Q squared or Q? For autosomal recessive, it will always be Q squared. Please know that unless they say the autosomal recessive gene, but if they say incidence of disease, frequency of disease, Q squared. Epigenetics, we clearly did Prader-Willi versus Angelman. Happy puppet was Prader-Willi or Angelman? Happy puppet was Prader-Willi or Angelman? Angelman. Prader-Willi, paternal, P for P, paternal intact, maternal silence, naturally, right? Paternal intact, maternal silence. 3 types of mutation: deletion, methylation and silencing, and uniparental disomy. So methylation and silencing in Prader-Willi should be paternal or maternal. Methylation, silencing or deletion for Prader-Willi should be paternal or maternal. Paternal. So Prader-Willi and Angelman are clearly distinguished. Look at this. This is what I asked you. Methylation, silencing, or deletion for Prader-Willi is? Methylation, silencing, or deletion for Prader-Willi is? Somebody said maternal, wrong, please. Prader-Willi, methylation, silencing, and deletion is paternal because naturally in Prader-Willi, paternal is intact and maternal is silenced, right? Yes or no? Or am I messing up? Yes, correct, right? Again, give me a second. Prader-Willi. Yes, so Prader-Willi, paternal intact. That's why there are 3 mechanisms. Prader-Willi naturally, paternal intact, maternal silenced. So there are only 3 mechanisms. You have to knock out both, right? 3 mechanisms: uniparental disomy, methylation, and silencing and deletion. Methylation, silencing, and deletion for Prader-Willi should be paternal or maternal. Methylation or silencing and deletion for Prader-Willi should be Paternal. Prader-Willi, paternal intact, naturally, and maternal silence. But you have to knock out both to get Prader-Willi syndrome. Happy puppet is Angelman. Tumor suppressor and proto-oncogene, clearly no example. Growth factor receptors, RAS. When is RAS active? Bound to? When is RAS active and bound to? GTP, MYC, they're all growth factor, they are signaling, they are all proto-oncogene. Proto-oncogene are green or red, inherited syndrome. Proto-oncogene is green or blue. I'm seeing different colors. Proto-oncogene is green, tumor suppressor gene is blue. Absolutely wonderful. Then clearly, in proto-oncogene also remember, so proto-oncogenes are growth factor receptors, RAS, MYC. Whereas tumor suppressor genes were caretaker and gatekeeper. Example of gatekeepers? Gatekeeper example, tumor suppressor gene, RB and P53. Caretaker, mismatch repair, and BRCA1, BRCA2. Then also we did chromosomal translocation. Remember, chromosomal translocation are fusion gene under strong promoter. CML and PML, AML, M3, they both are what? CML and PML, AML, M3? They are fusion gene, whereas Burkitt's and follicular gene under strong promoter, right? Remember, in proto-oncogene only we did these example. Proto-oncogene means either hyperactive protein or gene amplification or gene overexpression or these chromosomal translocation. Chromosomal translocation were clearly two. So this is what Philadelphia chromosome is, fusion gene or gene under strong promoter. That was an example of CML was an example of fusion gene. Then tumor suppressor gene, clearly distinguished from proto-oncogene. So if I say P53, don't call it a proto-oncogene. What is P53? What is P53? Tumor suppressor gene. You have to knock out both the alleles. You inherited syndrome, you inherit one mutant allele, but you have to knock out both the allele for tumor suppressor function to be lost. The second allele of the same gene and the same cell should be lost to lose that tumor suppressor function. So BRCA1, BRCA2 have breast and ovarian tumor because the second hit occurred in those cells. Loss of heterozygosity, is a tumor suppressor or proto-oncogene? Loss of heterozygosity, is a tumor suppressor or proto-oncogene? Tumor suppressor. RB, tumor suppressor. P53, tumor suppressor. NF1, tumor suppressor. BRCA1, BRCA2, tumor suppressor. APC, tumor suppressor. MLH, tumor suppressor. Clearly distinguish these two. I wish you good luck, and this will be posted on Canvas. You're welcome. I wish you good luck tomorrow. Is it tomorrow, right? Any question? I hope we ended on time. Well on time.
Thank you, Dr. Bog. Can we also get the presentation that you went over today, please? The document?
Yes. I shared it with your class rep. I hope she shared with you. And I'll post it on Canvas too. I shared with Shireen.
Okay, thank you.
But I'll post it now. But I shared before the session only, okay? I'll post it now on Canvas, okay? No worries. Good luck, everyone.
Recording